Perfect, thank you. Okay, so glad to be here today, everybody. Glad that it also worked out for me to just be here in person to do this. For anybody who hasn't like formally met me, my name is Alexis. I am going to be a fifth year PhD student now, starting this semester in the lab. So I started back in 2019. So today it's my turn in the rotation, and I do have a lot going on research-wise, but that story is still like forming. So I decided to present on two very cool papers that I needed a reason to read, and so this presentation seems like a good reason to read them and then like share what I learned with you all, because I do think they're very interesting, but they're quite dense. So the topic is advancements in human bionics for restoring motor function. That's what we're going to talk about specifically. For the overview, I'm going to look at two papers. Paper number one, title, this was published in Science Translational Medicine, Improved Control of the Prosthetic Limb by Surgically Creating Electrode Neuromuscular Constructs with Implanted Electrodes. Paper number two, Walking Naturally After Spinal Cord Injury Using a Brain-Spine Interface, and this was published in Nature. So both papers have to do with implantable brain-computer interfaces or body-machine interfaces of some sort. And then at the end, I'll do a little analysis. But for each paper, in order, I'll go over who the team is, who worked on the project, what the hardware, software is, and then what the whole system looks like. So for paper number one, primarily done in Sweden, this is an e-Opera osseo-integrated rod hardware system, so transcutaneous EMG classification to control a prosthetic arm. And then paper number two, Switzerland, cortical implants, plus a spinal cord stimulator, ECOG classification, and this is a central nervous system interface. So all of that will be explained in more depth as we go. I'm going to take a sip of coffee periodically. Okay, so paper number one, the e-Opera returns. If anybody who has actually been in the lab for 3 years ago, I gave a presentation on the e-Opera, and I was interested, or e-Opera, not sure, but I gave a presentation on 3 different papers that have looked at human studies of this technology, and now they published a new paper that I'm very interested in because of the way that they formed their neuromuscular interfaces. But like I said, it was published in Science Translational Medicine, and it's a return of the e-Opera. The e-Opera is an osseo-integrated implant. It has electrodes on the inside of the limb that are making direct contact with normal muscles, regenerated muscles, and then there's a transcutaneous rod to which the connection is formed with the myoelectric prosthetic arm, robotic arm. And in this case, most osseo-integrated rods, I think I've only seen in transhumeral amputees. The reason being that these folks have like the least residual limb, they have the least residual muscle to like hold on to these heavy robotics. And so these types of implants are intended to like extend the skeletal system and provide more mechanical support. But it has that added benefit of being transcutaneous. So now we have access to the muscles inside to use as signal sources. But before I go into that, talk about the team. So for this paper, this is from the Center for Bionics and Pain Research. They do a lot of the e-Opera research in Sweden. Some primary people, Jan is the doctoral student who, primary author, probably wrote the bulk of the manuscript, analyzed the data. Dr. Sasu is the hand surgeon, so he's probably the reconstructive surgeon who did some of the implants. Usually, Oskar Ajman is the surgeon who works on some of these projects, but they're both busy and it's good to expand the specialty. And then Dr. Ortiz Catalan is a professor at Chalmers. He specializes in pattern recognition for controlling myoelectric prostheses. And some other folks who worked on it, 2 postdocs who both specialize in the hardware side, 1 other doctoral student, and then Dr. Richard Brannemark, who is the founder of Integrum, which actually is the company that produces this technology and produced the osseo-integrated rods. He was, I believe, in Austria and has now since moved to MIT to work with you on lower limb stuff, or maybe upper limb. So a little deeper look into the hardware. The e-Opera, like I said, osseo-integrated. So the surgery, the implantation happens in multiple steps. First, you get your actual osseo-integrated rod, the part that actually extends your humerus bone to make it longer, stronger. What's great about bones, muscles, nerves, they all have the capacity to grow, which is very cool about the periphery. So the bone is going to integrate with this rod. So you wait 6 months, wait for that to heal. Then you add the abutment, which is this nice little thing that this is the actual transcutaneous part. So this is where you're now creating a permanent portal to the inside of the limb with the outside world. The primary purpose is like structural, so you want it to be a strong support, but there's also that ability to make electrical connections. So if you're able to tolerate the abutment for a certain amount of time and you don't get infection, you're like able to wear your limb fairly well, I think like Johnny, who works with the APL a lot, he also has an osseointegrated rod. He wears myobands, like Circus EMGs, to control his prosthesis while also making use of the rod. But in this case, and in what they do at this center in Chalmers, they also do implanted electrodes. So rather than recording from the surface, which can be unstable and may need lots and lots of recalibration every time you use it, they use implanted electrodes, which are supposed to be stable, very close to the original signal source, and also you're not like filtering out a lot of signal from skin, fat, etc. So having this transcutaneous connection is great because you can get really good signal sources. So that's just the hardware. There's a lot of interesting stuff going on with the musculature. I have a video that'll explain that a little better. But the hardware component, very straightforward, actually, very simple. Then when it comes to the signal processing, still the hardware, the actual like processing hardware is on board. It's all in the arm. This is from a 2017 paper. This is directly from my presentation 3 years ago. They are still using this same setup where they have a signal processing unit in the arm that has 3 major components, 3 major boards, one for neurostimulation. So this is bidirectional. You can record and stimulate. You can stimulate nerves directly. Mixed signal processing unit and a prosthesis communication and control unit. These 3 units have not changed. The hardware has probably made some updates in the past 6 years or so. But very similar, and these are the original specs for, this is called the ALC, or Artificial Limb Controller, and the specs are very much the same, except for one major thing, the channels. So originally, when I presented, they are recording from bipolar EMG patches. So the small electrode is put on the surface of the muscle. It has 2 contacts for recording a bipolar EMG, which we think is going to have like really good, low noise, be good for signal processing, and they have 8 of those channels. Well, in this case, the idea is that this individual is having a lot of reconstructive surgery, and we want to return as many degrees of freedom as possible to them in controlling their limb. So instead of 8 channels, we have 12 channels, and we achieve that not necessarily, I mean, obviously they probably changed out some of these boards a little bit in terms of like whatever, op amps they had on board. But in this case, they're doing 12 monopolar channels. So overall, the circuitry shouldn't have to change too much. But EMG monopolar data were sampled at 500 hertz with a 16-bit resolution and online high pass and notch filtered at 20 and 50 hertz respectively. So that's what they say in the paper is kind of the main signal processing element. But this is what all the hardware looks like. For the actual software, BioPat Rec, yes.
So yeah. They mentioned, since it is a bunch of electrodes, like how long those could last.
Oh, that's a great question. How long they last? Well, I'll tell you for this study that they look at this individual out to 2 years. That's quite long. I think that I would have to look through my notes to see what the longest human study I've seen is. But like the standard is 10 years. You want to feel confident that it should last about 10 years because that's the standard for implants. That has that tested. Has somebody like had to get theirs replaced after 10 years? I'd have to look that up. But that's a great question.
So is there a high rejection rate of the implant itself?
I do not know. That's a great thing to look up. But I will say that in terms of like, let's say rejection aside, in terms of the inconvenience of having a transcutaneous connection constantly in a small location is much less of a problem or a discomfort than having to wear a whole lot of sensors and a heavy socket to support a robotic arm. So I would say like, even if there is some discomfort, I'm sure they measure that. I'd have to look that up. But I would say in terms of trade-offs, they definitely consider that. And it seems like a good trade-off. And this person had great outcomes. So software, BioPat Rec, very old software. This is kind of what is on Dr. Ortiz-Catalan's resume as one of his big contributions during his doctoral and postdoctoral work is pattern recognition of these EMG signals for controlling the arms. And his software is all open source, and they always say they're using BioPat Rec. They've been using it for probably 10 years now. So I think that at this point they have a wealth of libraries and literature within their lab to use this very successfully. Some of the things that they were looking for. So from the channels they were recording from, they had a range of tasks, just general movements. Can you turn your wrist? Can you move individual fingers? They were looking at tasks like that. They also had some daily activity tasks like pouring a cup. And I have videos of that. But what were they checking every time he did those tasks? They were looking at in both growth movements, which are like those big arm movements, and single finger movements, which was actually moving individual fingers. Accuracy offline, eventually. So again, so they measured almost weekly, like several times a week or several weeks for a year after surgery. And then there was a long break. And then he came in like 2 years after surgery. So, so that's what these measurements are on the x-axis, and then on the y-axis. So accuracy offline, pattern recognition looks pretty good over time. Completion rate for motion tests, so that's like the percent of tasks complete or the percent of attempts completed. Just tracking and looking for like a linear rate increase over time if they can find that. Completion time for motion tests, just in seconds how long it takes. And then accuracy for motion tests. So this is when it's fully real time and he's controlling it. What is the accuracy of the classification based on like what he's attempting to do and what the classification is saying he wants to do. So that being said, the accuracy after 2 years, scores actually kind of low, and they point that out, that the accuracy is like 75% in terms of classification. But they say in their discussion that even though that seems fairly low, the individual, or cyborg number one, who is a middle-aged man, he said that this was still way better and way more intuitive to control, way easier to control at home. And the reason that they say is because spurious misclassifications are common in decoders, but do not translate necessarily to noticeable errors during prosthetic control because classification output is continuously overwritten and low pass filtered by the motors in the prosthesis. So that's kind of interesting. I thought that that was like a standout point to me, like the difference between what your classification algorithm is predicting in a millisecond level versus what's actually being translated at the end of the lever, at the end of the robot. Okay.
Alexa?
Yes. Yeah, go ahead.
Just a quick question. Can you explain a little bit how they got that measurement again? You're saying it's the difference between what they actually do and what they were trying to do? How do they know what they were trying to do?
Oh, right. This is like they say bend your elbow, and every single sample is telling you at a given sample what is he trying to do. Bend elbow, bend elbow, bend elbow. Oh, maybe something else. Bend elbow, maybe something else. Like that's what they're saying is happening, is that the classification algorithm is generally getting it right, but sprinkled in there every millisecond or so, it's predicting something wrong. But those spurious misclassifications, they say, are like being filtered out by the overall system.
So this is like prompted, and they're seeing the output of the hand as they're doing it?
Well, they're measuring that graph in particular, this accuracy percent, in the accuracy of the classification overall. So for all seconds, all of the seconds that it was running while motion was being classified, whether rest, move, whatever, how accurate was the real-time classification algorithm in the moment? And every second, what's the probability that it's going to be right? 75%.
And what do they consider the big difference between D and like A, for example? Is that just because like the real time
offline versus real time? So I think that offline they train it off. They probably like let it train for longer. This is a little in the detail, so I will focus big picture, but I'll say that has to do with like this is just the offline and they train beforehand versus real time, which I bet they have quicker windows, quicker update windows.
Oh, I see. Okay.
Okay. All right. That was a great question. All right, so making sure this is off. All right, so a little bit more about that surgical element, about why I was interested. So they're going to dive into this person originally. This is what the residual limb looks like. You've got parts of your bicep, part of the tricep that have been cut off because of the trauma. Those nerves are now going to be targets for reinnervation. So the nerves that used to travel all the way down to the hand can now be surgically manipulated. Some of those some of these residual muscles, you can just quickly denervate it like this short head biceps, and you can reinnervate it with the ulnar nerve. Or you can take a chunk of muscle from somewhere else in the body and wrap it around the cut nerve ending. So these are nerves that originally were not doing anything. They were just hanging out in the residual limb, and now they're being given new muscle targets. Some of those muscle targets already existed in the residual limb. Some of those muscle targets are being brought in from outside in the form of these free muscle grafts. Those free muscle grafts are being taken from the leg. The vastus lateralis muscle is a common target, so it can spare a lot of tissue. So now they've got some normal muscle, some residual muscles that have been reinnervated just through putting the nerve to that muscle, and then now they also have free muscle grafts. And all of these act as signal sources. And one other thing I want to point out really quickly is that what's interesting about having these mixed signal sources, having native muscle, targeted reinnervated muscle, and free muscle grafts, 3 different types of signal sources, this is exciting. And it's also interesting because what they demonstrate is that they use, like I said, monopolar electrodes. They use epimesial patches in this case. You cannot put an epimesial patch on top of a free muscle graft because they're not vascularized. They're just like soaking up nutrients through diffusion. So if you put a patch on it, you're gonna kill it and you'll get no signal. So in order to have successful reinnervation, you have to actually use intramuscular electrodes. And there's always some kind of debate about intramuscular, epimesial electrodes. They had some very interesting outcomes by using a combination of the two, and they would also, in some cases, put both an epimesial and an intramuscular electrode on the same muscle to look at the differences.
Alexis?
Yes.
You mentioned before that they're doing both recording and stimulation, or they're capable of doing it?
Yeah.
What are they stimulating?
They do not stimulate in this paper, but this individual does have a stimulation cuff on one small fascicle of the median nerve. I think it's probably one of the thumb actuators. I think that they're hoping to like get some kind of like tapping stimulus. They have written other papers where they do that with folks who have even more cuffs, but they don't stimulate in this paper particularly, which sensory stuff is cool. I think a theme of some of this work though is that like sensory stuff is still kind of unknown, new territory, very new. So we're kind of more focusing on some of the EMG aspects, although there's some sensory stuff maybe. No, there's no sensory in the next one. But the point is that the hardware is capable of being bidirectional. Okay, come on, there we go. Okay, so here's some example of control. They're demonstrating that you're getting single finger actuation, which is great. I think maybe did it skip the, no, that's in the next video. Here we go. The next video has a little bit more movements. So some demonstrations of doing daily activities, which I think this one is really interesting. I like this video in particular because you can see if the resolution of my video is good enough, which luckily it looks like it is right now. You can see muscles twitching in the residual limb, kind of like independently. It looks unnatural in some cases because like, you know, one part of the bicep will flex and then another part seemingly independently. That's because there's a whole bunch of stuff going on inside that residual limb. That's incredible, like regeneration has happened. And it's cool to actually see the muscles twitching that were reinnervated in the bicep. I also thought that was cool. That was a demonstration. He did a full 360 wrist rotation if you were watching. And I fully thought they were gonna do it here. I was like, oh, they're gonna demonstrate how he could just fully go 360 and like fully screw that in. They don't, it's a big tease and he puts it down. So I was kind of disappointed by that because I think that's like a cool element of cybernetics. Cyborgs have extra capabilities we could explore. But generally overall, they do more scores, more behavioral scores that I don't show here because I'm less interested in that. The next.
Could you play back that part where he was like holding a ball and swinging his arm around?
Yeah. In this case, I was kind of looking to see how much elbow control there was. I didn't see much, I thought, in some of these demonstrations.
Interesting. It's hard for me to tell, but I was trying to see how much of it was like shoulder motion versus like actual arm was moving. It's hard to tell when it's going fast.
Yes, and these are also only select videos. So,
yeah.
Oh, that's another great thing about all these human trials is that they have lots of video content. But now just to look at raw EMG signals, which I'm very interested in, they have the 3 different types of signal sources I mentioned. So on the top row, this is EMG over time, up to like a year, and then 2 years out from surgery. This is just unreconstructed long head bicep, normal, you're saying flex the elbow. That's what the bicep normally does. And recording monopolar EMG signals. Looks good, even looks like it increases on average. I have a couple theories as to why, but overall it's pretty straightforward. Doesn't seem to get worse, that's great. The second category is native muscle that was left intact, except it was denervated and reinnervated by one of these big hand moving nerves, the ulnar nerve, which flexes your ring and pinky finger. So in this case, native muscle reinnervated by the ulnar nerve at week 16, they say, flex your ring finger, and this is what you get. And over time, up to a year, they start to get really fantastic examples of reinnervation where they're seeing much, much larger signal to noise ratio at a year out, to the point of seeing like 3 millivolt amplitudes in this reinnervated muscle, which is fabulous. And then that's compared to the free muscle grafts, which are also reinnervated. And I actually don't know if the difference, who's epimesial, who's intramuscular, but we know this one has to be intramuscular, which is that the free muscle graft, same thing at week 16. This is median nerve, so flexing your index finger at median, at week 16, nothing. And then by 1 year out, very distinguishable signals, I would say, at about 100 microvolt amplitude. So that's like, I mean, that's low, but that's pretty good. That's also pretty low compared to what I've seen reported from the folks who do those free muscle grafts in Michigan. They've gotten some pretty high values, but that might have to do with differences in filtering, differences in recording setup. So, but the fact that the ability to detect signal, that's great.
Do you have a guess as to how much the type of electrode affects the results rather than just the type of surgery?
Oh, I think the type of surgery has a way bigger effect, for sure, yeah. And I think like that's why I'm like, I don't know, it would maybe be interesting to look at the differences, and I'm sure if we dove deeper into the weeds on this paper, we could find out. But I do think there's a lot to be said about how much damage has been done to these free muscle grafts. Like it's such a small amount of tissue versus something that's being mostly left intact. It has a lot more capacity. So I'm not surprised. I think the drastic difference is interesting, and I'd like to see how can we manipulate that through more experiments with animals, probably, because you can't do these kind of controlled experiments in humans as easily. But again, this is just more of like a linear look at looking for like, is there a weekly increase in signal to noise ratio that we can detect that is like fairly accurate? So these black lines are just their linear regression. Yeah, I mean, increases in signal to noise ratio over time, that looks good, that's validating. I think what's a little bit more interesting is looking at, this is good for me, like this information about the signal to noise ratio, the quality of the EMGs, this is useful to me. I think what is more novel about this paper though is like how many degrees of freedom they're able to achieve in this person who has such a small residual limb, and how through reconstruction and clever engineering, they can give him an improved quality of life.
Sorry, I can't read the charts, but there's different numbers. Are those representing different muscles or?
Oh, yes. Yes. Okay. So again, so this is actually, so in this previous, let me just go back. So in this previous graph, this is a single electrode over time for each one. And in this case, now it's like, okay, what was happening at each electrode? Well, in all of our naive muscles, we expect flat. All of our reinnervated muscles, you expect increase. And so these are just the reinnervated native. And then the
So they just have
muscle.
different electrodes, number of electrodes for the different
Yeah.
setups.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 monopolar channels. This is all the channels.
Oh, they were doing them all simultaneously,
Uh-huh.
even though they're like different muscles, presumably. Different muscles. Interesting.
So yes, they increase the channel number without doing too much to change the hardware. So they maintain those benefits of like the offline control they've been able to achieve. And they use a combination of targeted muscle reinnervation and RPNIs, free muscle grafts. I'll say that the authors of this paper call them free muscle grafts, but RPNIs is part of the surgical literature to call those little muscle grafts. So using a combination gives good prosthesis control. Chronic monopolar electrodes produce good SNR in a lab for more channels. So that's just really interesting in general because they were like nervous about this. They were like, oh, we can't give up being bipolar, but you know, it does have trade-offs in terms of space, freeing up some channel space. So that worked, and they're satisfied with that. And then the other thing they said was electrode placement in surgery is not optimal. So when you're doing the reinnervation surgery and you're figuring out where to put your electrode, you cannot just know where the best place is. Like obviously, you move it here, it'll be a little higher, here it'll be a little lower. Generally, you would find the best spot, suture it there, and like that's the best spot for the highest signal. At the time of surgery, those muscles have just been defunctionalized and they don't work. So stimulating them is a little like trickier, and you're not really looking at anything physiologically relevant. You don't know where the best place is going to be 3 months from now. So you have to guess, you have to use intuition. With those free muscle grafts, pretty easy. You put it in the middle, you have no choice. But for other things like those targeted reinnervated muscles, where do you put it along this kind of large muscle? And also, how do you avoid crosstalk from a nearby muscle when you're using patches on the surface? So you're going to get crosstalk when you use epimysial. Epimysial does less damage. Lots of trade-offs. But what I really took away that I thought was interesting was that they pointed out specifically that they would benefit from knowing, if they could know in advance what this muscle is going to be, you know, where would be the best place to put this on this muscle 3 months from now. To know that in advance would be helpful.
Do you know if there, or do you have an intuition if there's like some sort of measurement problem, like meaning where you put the electrode ends up affecting the recovery, and therefore, like almost by definition, it won't be an optimal spot because it's like impeding the recovery?
Yeah, that's totally possible. That's why I think we need to model it, and we need to be able to like add things to our model, including like physical blocks. I think that's like something we would have to do in animals to figure out, like how big, what is the size threshold for a patch electrode? How many holes do I have to put into my patch electrode to make it porous enough for this muscle to survive? these are things that we can only answer in like animal studies.
So I'll just make a tiny interruption. That's exactly the nature of it. I don't know why you didn't say it, but it's the nature of Alexis and Kiara's PhD to look at issues of perfusion, innovation, muscle health, production of motor unit, and so on. You know, it's, as she has said more than once, some of these questions can only be answered in animal model, try out and bury and all of that. And
Paper number 2 is going to build on that theme of like, this is a bigger team, this is a little bit more developed tech. I think that this paper is very interesting to me because it's doing a proof of concept for me. I would love to be able to do this in rats, to watch muscles reinnervate slowly over time in rats, prove we can do that, and start playing around with those variables we just discussed. But that's a little harder to do. You don't see osseointegrated rats for a reason. You know, you have to think a little more carefully about a model. Imagine trying to collect this data from this man and you have to trick him and you can't tell him what he's doing, but you're trying to collect data from him. That's what it's like working with rats in these scenarios. And so you have to be a little bit more thoughtful to try not stress them out, they get the reaction you want. So I'd love to replicate this in rats. Yes.
I have just like a question for you. If you had to speculate, why was this work published now versus like 5-10 years ago? What's like the jump that caused it to be feasible now?
So let's say 3 years ago when I was presenting, they had been showing just targeted muscle reinnervation. I think that what makes this paper novel is that they added RPNIs. They added these 3 muscle graphs, and they're kind of some of the first to publish on it outside of the Michigan group who originally did it. And they're demonstrating how much the spatial resolution helps, how much it gives you like more channels to record from, give you better degrees of freedom for a transhumeral amputee, whereas the Michigan group only shows data from wrist dislocations, from amputations at the wrist. So like that's a very different scenario. And I really, I think this adds a lot in terms of proof of concept that the surgeons in America that I listen to, they kind of battle like, I do TMR, I do RPNIs. I've always said that it should probably be a combination because that's what they all end up saying at the conference anyway. And these guys demonstrated. They were like, we used to only do TMR. We decided to add RPNIs. Look at how many channels we were able to achieve.
the crude answer though is building the team and getting the funding.
So, and this is really, it all ties together. So this next paper is in Nature. This is going to be reinnervated muscle, very cool. You shouldn't let me speculate because that's what I like. But this paper shows what reinnervated muscle studies could become, what the next level is, because these guys have been working for a really long time and done a lot of interesting stuff. So walking naturally after spinal cord injury using a brain-spine interface, published in Nature. Gorgeous, gorgeous figures, beautiful figures. I think all of us should read this. I have like a printout copy. The paper itself is fairly small and the supplement is quite hefty. So like I'm barely going to scratch the surface of this paper, but it's a great read, especially even just the small article part in terms of like, this is an N of 1. This is a case study of a brain-spine interface, autonomous brain-spine interface. This person is able to like use this whole system fully implantable on their own to go from paralyzed, spinal cord injury, cannot walk on his own, can now walk on his own, knowing how to like program, turn on his BSI, he can use it outside of the clinic. And so that's why this paper is in Nature. Not only does it show that, it shows it robustly. There's tons of beautiful figures. But first, before we look at the figures, let's look at the team. So this is team Switzerland, France. I would say it's mostly Switzerland, France. There's sprinkles of other folks in there, even the US. Henri Lorach is the postdoc scientist who led the team, led the manuscript development. His PI is Grégoire Coutine at EPFL. So this is in Lausanne, Switzerland. And their team works on NeuroRestore. It's restoring the walking ability through spinal cord stimulation of paralyzed folks. They work with a surgeon, Jocelyn Bloch, who is a surgeon who works with them. She does some of these implants. And then now for the first time, they're doing their spinal cord stimulation to restore walking. They've done that before, but they usually control it from externally. They use video, they use EMG. This is the first time that they restore spinal cord stimulation walking using a brain implant. So this guy is leading the team that does the brain implants. So there's a spine team and a brain team. And these are all part of the team. Much bigger paper. And also these people are also part of the team. It's a huge team. There's 34 authors, and it's split up roughly like this. You've got EPFL and Lausanne and NeuroRestore, Lausanne, I guess, and they, big team, lots of postdocs. Medtronic for the spinal cord stimulator. That's just an implantable pulse generator, and who do you think helped out with that? There's a surgical team that's working for this individual, making sure that they're healthy, that their nervous system is healthy throughout all of this, as well as the implants. And then Grenoble is where the cortical tech is developed. They were the ones who first published on these ECOG arrays. that the Neuro Restore team now wants to use to control their spinal cord interface. So look at what that looks like. We have electrocorticography, or ECOG, being recorded by a fully wireless bilateral implant called Zbinden. It has 64 channels, and it's great. I have pictures in a second in the hardware. Spine is an implantable pulse generator. Yes, 64 channels each side, left and right hemisphere. The recording part, well, you'll see. But this is for ECOG, which is sensing the desire to walk, and then the implantable pulse generator is stimulating the spinal cord, generating the walking pattern. This is what that looks like. What's cool, this is the antenna for communication, wireless data, and power transfer is occurring through the skin because the antenna is subcutaneous. But the actual ECOG sits on top of the derma. So the actual body of the device is level with the skull, and you get kind of a nice seamless implant. So there you go. So this is what the Zbinden tech is. They have multiple papers on it. If you're interested, I encourage you to read them. But the main takeaway is that they can wirelessly, through these headsets for the inductive transfer, they can acquire ECOG signals from, they're targeting M1. They're targeting the areas of the brain that move the legs. And this is kind of what that looks like. This is high depth. I'm going to do some skipping. But the main point is that they turn the system on. Okay, so now we're going to start recording ECOG. What do you want to do? What is your brain saying to do? They get this spatial element of all the 64 electrodes are going to light up differently, depending on what part of the body you're thinking of activating. And each signal itself in time is going to have spectral features that will say something about which muscle you're interested in moving. So taking those spatial, spectral, and temporal features from the ECOG, they have a very fancy algorithm. And I think I understood it. Let's see. There's 2 separate predictions. It's running real time. I have it highlighted if you want me to read it later. But just the algorithm itself is specifically based off of lots of stimulation patterns that they have developed already in their animal studies. I'll talk about them more later. But they have patterns of stimulation in the spine, which will generate walking. It's just kind of a pattern. It's repetitive. It can be done. It's been demonstrated before. But now we're doing it with this brain. So they have to have a real-time algorithm. That's another reason why there's a lot of people. I'm sure that real-time algorithm, there's a lot of software engineers on here. But then once the classification is done, once we decide I want to walk or stand or pick this leg up more than this leg, then it's just a matter of delivering the stimulation. It's a Medtronic implantable device, well validated at this point. Stimulation programmer, just like you would expect in someone with Parkinson's to be able to program their DBS. This is what it looks like. These are beautiful, beautiful figures. One of those people on that team, his whole job is figures, which was a very interesting thing to find. But this is an example. So I'm sorry I forgot this guy's name, but Cyborg number 2, and I say that as a term of high endearment. We're doing motion tracking as the BSI is off versus on, and looking at the ability to elevate the knee. You'll notice that he has some capabilities even with the BSI off. He's been doing rehab with these folks for a really long time, and they've demonstrated multiple times that like doing these spinal cord stimulation is actually improving his abilities to move without any kind of interface. Like it's helping him recover from his injury. But that being said, still not perfect. He can't take steps. He can't walk without the BSI, which is demonstrated when they turn it off mid-motion and as he's walking. So it's planned. They tell them they're going to turn it off, but they don't necessarily say when, and they're interested in seeing how the brain adjusts to that, and also like seeing these attempts of trying to walk but not getting any EMG activity. So this is the brain activity saying left, right, left, right. This is the decoding probability, right, the classification. And this is the actual EMG activity of those muscles, and you see no activity. Ankle trajectory, that is from motion capture. So this is a basic overview again. For like more details, I highly encourage you to read it. The actual article itself is short, and the figures are beautiful. You can learn so much. But I want to show you this video from the supplement. So he was doing so well in that laboratory environment that they were like, well, he was like, I want to use this at home, obviously. Like, it's doing so well on crutches in our environment where I've got all these suspenders. I want to be able to use it at home. So they went about developing a walker setup. So in the lab, he's got recording headset. He's got a processor in his backpack. He's on crutches, and he's got a spinal cord stimulator. In this case, now he also has to do some calibration and have some tools to like to start up the recording to make sure everything's calibrated, good to go, and then also a walker for safety. If you're going to be out in the real world, you're going to need a little more assistive devices, and walkers are much more easier to manage than crutches. So let's go ahead and there's done. So you can see that like the walking is kind of happening in stages. That makes sense, right? This is also being sped up 3 times. It says that on the video. I'm not speeding up anymore. But so it's not perfect, but that's going from you can't stand or walk without it, and you can with it. I think that it's a fantastic demonstration. Both this and paper number 2 demonstrate that like a very thoughtful, big team behind very intelligently designed surgeries plus tech, you can get really great outcomes for folks at home, like complete autonomous use. This case, I think it's mostly enabled by the wireless aspect, the fact that both the recording and the simulation is completely wireless. The most important elements are actually housed inside of his body, and then there's a whole bunch of computing sensing that goes on outside. If there were any transcutaneous wires for anything to do with the central nervous system, I mean, no way. That's way too dangerous. I mean, like in terms of risk of infection to the cerebrospinal fluid. So, so anyway, I think that what was great about this demonstration of the autonomous use is that like previously he had been performing in those other forms of spinal stim where they're using video and motion capture to control his walking. Now in this brain control, he's able to do things he couldn't do before. So in previous, and that's what the STIMO means, like he's been in previous studies. But he can do ramps, he could not do ramps before. He can do stairs, he could not do stairs before. And then they have this kind of crazy like mixed terrain thing they had him walk across. And very interesting, like to get all the data from that and look at its adaptability real time for him out in the world. They did some tests with crutches, which is kind of cool. Obviously that was probably like not doctor recommended, or maybe that's the doctor standing right there. That could be Jocelyn, so who knows? But here's a picture of the team. So again, this is Henri, who led the team, Dr. Lorach and Dr. I jumped out of my head, it's French, Courtine, I think. And then this is Guillaume from France, who led the team on the brain implants. Major conclusions. A fully wireless brain-spine interface is achievable in humans with modern implants. That was cool. I think you should all read it. Further validation of the concept that epidural spinal stimulation can improve outcomes over time. So there's a regenerative element that by introducing this stimulation, they're actually improving his outcomes even when the device is off in his PT. And there's an impressive stability of signals. They talk about how often they had to like reconfigure the ECOG. It's very rare, and they discuss that like that's configuring like what part of my brain moves my ankle. They do that all in a couple of big sessions, but for 2 years, I'm sorry, not 2 years, about like 18 months maybe, he's been able to use this autonomously without having to recalibrate the ECOG classification. Compare and contrast.
Sorry, before we do that,
Yes.
a couple questions about that. So, in terms of the ECOG implant, where were they targeting? Was it just primary motor cortex?
Yep.
Okay, so it was like muscle intention, not not more abstract movement intention necessarily.
I forget how they like,
So
they did
this was done a
pre-localization.
decade ago by 2 or 3 of our PhDs in other things. Or like, anyway, and Nathan Cohen and the surgeons, very generation before, so it goes back 10 years. There's nothing like, I mean, M1 area is about, entire finger control about 4 millimeter cube. So the electrode size is half a centimeter.
Yeah.
Centimeter.
Yeah, so they are also
This is
targeting.
kind of like a
premotor.
network, you put
Yeah.
a grid of electrode on the subcortically on this, and then derive a reasonable premotor signal. So it does show up in a more slow motion, low frequency signal. But again, I don't want to distract you. You can fill in the gap.
The chart area, yeah, motor.
Yeah, I guess primary.
It's a motor, supplementary motor, S1. They all basically code something. You go to microelectrode, you'll find something.
They also
Then
do
electrode
like,
array, it's a premotor planning. So it's a graph of all of that. And then if you're lucky, the luckiest part is that our students are lucky because most of the patients who come for this are epilepsy patients. So it is what's called temporal lobe and usually on the left side. And what you need is central sulcus on the side that is contralateral.
Yeah.
So therefore, if they get that right patient, they can cover it.
yeah. It expands the central sulcus a little. They have a supplemental figure all on that calibration.
And then the other thing was, like, for activating the muscles, do they need to, like, let's say they just want to stand.
Mhm.
Does it need to be active to do that?
Yeah.
As well? Okay.
He
So,
can do it without, but they actually use motion tracking to validate that standing is more stable, more natural with it on.
yeah, because you have to like
Because you're
more,
not using your arms for like crutch.
yeah, you have to like more actively activate the muscles to just maintain the posture. Okay. And then are they, I guess like the gate, are they just like predefining a certain frequency, or is it based off of like some measurement for them that
Like
they're
what's a normal gate?
reproducing? Yeah.
Yeah, it's based off of like tons and tons of data in normal humans, also neuro-impacted humans like him, because they not only have this very awesome hardworking cyborg, they have two other guys who work with them, so they have multiple forms of like, they generate libraries of motion tracking. They've been doing it a long time, which I'm gonna show too. They've been doing it for years.
Okay.
So yeah, so some of their normal stuff, you don't have a true normal for this individual, but you can extrapolate similarities between these papers. Both were n=1, which is kind of fun when I was talking to Sammy yesterday about one of these papers, and he was like, well, how many people? I was like, just one. And it's a good paper, and we're spending an hour talking about it. For both, it was over a year of data collection. And prior to starting that, they already had multiple years of data collection from other humans in both cases. Relies on implantable tech for both. Demonstrates at-home autonomous use for both, and multinational team for both. Did I miss any? What do you think? I think that's differences, I didn't really think too hard on this. You could be like really in the weeds about it, but I was like, all right, big picture differences was the interface. We have transcutaneous bidirectional peripheral, and we also have fully wireless unidirectional central nervous system. And then the other differences are mostly like, number of authors. So like number of authors, paper number 1 had 7 authors, 2 were doctoral students, 2 were postdocs. Everybody else is like a professor or a professional. And paper number 2 had 34 authors, 4, so even a smaller fraction, 4 were doctoral candidates, doctoral students, 6 were official postdoc researchers, and the rest are all like either professionals, they're working for the company that spins out this, the Wimagine, so they have a company, and also the calculation for spinal stim, they have a company, or it might be an institute called NeuroRestore. In Switzerland, I'm not quite sure how that works, but we'll call it an institute, NeuroRestore, where they work on this restoring walking through spinal stimulation. So lots and lots of people with 3 or more letters behind their name. And they also occur, they were both done in different places. I was interested, I was like, what if I went to go work here? Doctoral stipend pre-tax per month in Sweden is 3,025. In Switzerland, it's 4,662. And at Hopkins, it's 2,996. So comparing Sweden, not that different. We actually may have gotten a raise since I checked this. Switzerland looks great, but I also looked it up just for fairness and transparency. The cost of living in Lausanne, Switzerland is much higher than Baltimore. But I just thought that this was kind of, it's really just to engage you on the idea that think about where this occurred. What is their work culture like? I've had a lot of friends start doing internships in Europe, and they come away with the concept that like the work culture is different. The way things are done is different. So maybe we could all start investigating that on our own, considering going abroad if you're interested. And then
Mark could be going,
beautiful.
away,
I'm going to zoom through this because I'm over time and I want anybody who has questions to ask them. But so the one thing I'll say about the spinal team that I think made their team so robust, and this was very interesting and why they're in Nature, is that I also remember this paper from like 2018, I don't know, but it's the same PIs and none of the same authors. These are all totally different people, but all the methods are the same. They had full teams working out how to do all of this in animals long before they went on to do these human studies. And so they, because I was very interested long ago in looking at their rat setups. They had a full rat model with a spinal cord stimulator and motion capture and EMG recording. And they also figured out where these spinal hotspots were. They gathered a ton of data about how certain activation patterns cause certain walking patterns. And it's just, it's very interesting. Maybe I'll post this slide later, but.
So this next clip, I wanted to show during the meeting, but for time I skipped it, so I'm gonna drop it here.
The leading mind in cybernetics. He lectured at my medical school. You know the disaster at Macromius? Well, since then I've been working on an approach that combines cybernetics and regeneration. It sounds impossible, I know, but I have found an approach that will work.
And the reason that I wanted to include that video was, one, because I love Star Trek and I will find excuses to incorporate it into my work, but 2, I also think of science fiction as like a source of inspiration for our work. Regeneration and cybernetics, like combined, is a big part of what I want to accomplish in terms of like modeling, studying the mechanism at a more granular level in animal models. So yeah, I wanted this clip just to be like silly, goofy, get your brain juices flowing.
We're not going to watch Star Trek. It was just to be a little silly, but the idea is that to apply this to our work, in my work, I'm very interested also in how muscle regeneration can be leveraged to improve prosthesis control. So that was a big theme in paper number 1. And I specifically use rat models of muscle reinnervation to try to study those things. I have some acute models that I've been working with and validating for years now, which is to do the nerve coaptation versus the nerve grafting and to look at differences acutely. But my long-term goal is I want to track it long-term over time. I want to watch the signal to noise ratio go up in these rats chronically, wirelessly. I think that that's kind of why I think that these two papers go well together for me, at least, is that the one demonstrates the human need and like the need to apply some of what we learn in animals to the human case for like electrode placement, bioelectrical modeling, being able to take imaging data and make lots of predictions and lots of models before you go in and ever actually do any surgery. And that's just what I really well. I think that our lab has an opportunity to do really good data science and to like think about data management so that we can build good models that would then inform a lot of these bigger human studies, possibly even future clinical studies at Hopkins. We have the capacity, but if we have really good data and models to back it up, I think that that's going to just push us closer to humans potentially, if that's what we want. This was a nice break for me to read these papers. I'm glad I read them, even though I did it a little cursorily in order to
This
get
figure, if you see in the left one, was probably designed by you or initially started off used by
This was from
Connor.
Luke, and Luke was actually, one of his papers, this paper I grabbed this from was cited in the osseointegration paper. So yeah, I'm like trying to capture even like a small portion of what a much larger project could look like for us, but I do think that creating amazing models that then we can like leverage out to other teams would be my dream.
Which is, for example, the model/electrode array that is robust, developed by folks at Emory.
Yeah.
Is helping. And they, if you look at their paper, they've spun it out to half a dozen universities.
Oh God, we should ask Emory for a letter for the R1.
Sure, no, or even a co, depending on what,
Okay.
So we can take that,
Yeah.
Alex is helping, literally driving some development of an NIH grant. So this is what it is. It's kind of like teaming up. And, what I thought that, student culture is what, right? You take my instrumentation class and you and yourself or you and one partner, you're used to working with, maybe you have to submit separate reports so that you can be graded separately. And coming from exam, of course, all by yourself. And coming from there, grad student, you cozily talk to and work with somebody next door in your lab, Charles.
Well, it's a very collaborative culture. My friend who's in Switzerland now.
That is the difference. So what happens is that, and how many times I've brought up, get out guys, collaborate, reach out, bring in external, internal collaborator, but the inertia, perhaps a little bubble effect, you want to finish your work. But we have Tanguy here, we have at Brazil, Camille from Brazil. I mean, it is possible, European connections. Smart is helping organize a pretty good workshop where we're building the network. Some of you have for different things invited people, at least by Zoom was a very fortunate thing. So now we can really invite any of these people by Zoom and they'll give us a talk. And it doesn't have to be some famous PI, it can be a postdoc or a junior person who spun off and is starting to build a lab and facility. So this is where what I think selfishly, and I've used this, I know I go into cliches, I know I've used this cliche, that be scientifically selfish. You really should, because your self-interest is served, you'll be fine. But you have to make it clear to me and the rest of your team to make it happen. So what do I mean by that? You're a PhD, really, at the end of the day, write a really great paper. Because that spins off everything. It's like it fits your PhD, you will get award or recognition, and people will cite it, and it'll be part of the story. But it may take more than you. It will take a team. Okay, so that's one way to be selfish.
Well, I do want to open it up to the floor because it's 11:10.
Yeah, okay, sorry. See, that is my
questions, discussion points I've asked, also what you guys think of the transcutaneous versus wireless debate. I'll probably post this video somewhere.
We have a lot of these groups, resources at Hopkins, wireless, federals are there, med school surgeons are there, but it still happens. It's not a multi-institutional, international, so on and so forth. So, please open up such things. Okay, I open up for dialogue, schedule it if you like, or we'll stick to update of your projects. I think this is important to develop these kind of big paper career discussions.
All right, cool. Thanks for everybody's attention. I'm going to stop sharing now. And I do think that, and I can stop recording.